All right, I'm jumping in, minimal introduction. I'm just going to take you through these slides as quickly as possible. This is the last bit of information you would need to know in order to ace that test that we have coming up tomorrow or Tuesday, depending on what class you're in. So we're going to talk now about cells communicating with one another, and it definitely fits within this chapter because we've been talking about the cell membrane, the proteins that are embedded within it, carbs that mark it, and so why not talk about how those parts have to communicate with each other in order for you to get like a combined response. So cells, they work together to make things happen. They're not working by themselves. It's important that cells are able to communicate with one another. Everything that your body does depends on multiple cells working either in unison or in a sequence. Cell proximity has an effect on communication. Both plant and animal cells have some direct connections, meaning the cells are touching each other. And when this is the case, they can send messages back and forth via gap junctions, and that would be for animal cells. And then plasmodesmata are what the plant cells will be using for this direct touching and then sending of a signal. So one form of local cell signaling could be something as simple as one cell secreting a solute of some sort from its cytosol and passing it directly into the neighboring cell's cytosol. So just imagine, like with the plasmodesmata, these two plant cells are right next to each other, and one of them has a solute and it sends it or diffuses it into the cell that it is touching. Same thing would go for an animal cell, except for it wouldn't be plasmodesmata. It would be these gap junctions through which the solute is passing from one cell to the next. So sticking with this whole idea of local signaling, we see in the first bullet that cells can signal one another by excreting specific molecules to elicit a specific response. And the response that we're looking for is coming from the cell that is receiving those solutes. So these messenger molecules, they can be referred to as local regulators. They say it right here in this second bullet. Messenger molecules, local regulators. So they give us an example in the following bullet point about growth factors. So growth factors are able to work in this way where the message only has to travel a very short distance in order for the next cell to get the message. It's not something that has to come from, far away. So once the cell signals a nearby cell that growth is needing to happen, essentially the next cell readies itself for mitosis, and it does so by growing a little bit bigger before division actually happens. They call this type of local cell signaling paracrine signaling. So right here. Okay. All right, so we're looking here, figure 5.19 or 5.19. We see local signaling happening, specifically paracrine signaling happening. You have one secreting cell, so this guy here, they give you these little green molecules. Those are going to be the local regulators, and the secreting cell, it just lets these molecules go. And in the extracellular matrix, or in the space in between the cells, the nearby cell can pick up on it. It would take in these molecules, and then you would get the response that we just spoke about, or I just spoke about. All right, in this slide, we get another example of local signaling, but this one is a little more specific. So the book mentions local signaling within the nervous system, and your nerve cells, they don't quite touch one another. They have a space in between the signaling end of one and the receiving end of another neuron. So what happens is an electrical signal moves along a part of the cell called the axon, and that is what makes a neuron secrete molecules called neurotransmitters. The molecules, they fan out via diffusion into that gap space that I was just talking about, and they hit the receiving end of the next neuron, and you refer to that as the target cell, wherever the response is being focused. All right, so we're still looking at figure 5.19, but this time we're looking at part B of it. And so what we see is this is local signaling. They show you a part of the axon, kind of. You don't see the top part of the cell, but it would all be up like towards that way. And so inside of this neuron, we can see these small vesicles that are containing neurotransmitters, these circular vesicles here. So what happens is the cell is releasing those neurotransmitters, they hit the target cell, and that makes the signal continue to flow. Now we're going to shift to long distance signaling. Hormones are chemicals that are used for long distance responses. The endocrine system in animals, which is primarily responsible for the release of hormones into the circulatory system, and to elicit a response, a specific response depending on the type of hormone that's released. We'll talk about that more when we get into the nervous system, the immune system, and the endocrine system, so the hormone system. So there are a bunch of different types of hormones. They work in different ways. We'll talk about more specific hormones when we get there. But yeah, so we'll deal with that later. This is all this slide is saying to you. All right, so here is the third part of figure 5.19. So this one is showing you the long distance signaling portion. So you see that there is one cell, an endocrine cell here, and within it, it's releasing some type of molecule. That molecule has to travel through the bloodstream, and once it hits its target cell, it will elicit the correct response. But what you should keep your attention on is the fact that the original place from where this cell signaling is happening is the brain, and the target site is down in the pelvic region. So we can assume this has something to do with, of course, hormones, but it could be something to do with eating, or it could be something to do with puberty and growth. So either of those would work. So there's this scientist, his name is Sutherland, and he is the one who figured out how epinephrine works, which gave us great insight into how some of these other hormones could react, I guess. So what he suggested is that cells that are on the receiving end of epinephrine, they have to go through 3 stages. So first they have to receive the message, so that's what they call reception, and then they have to move the hormone along, that's the transduction piece, and then a response will be elicited, so that's the response piece. So we're going to go through each of these processes in the remaining portion of this lecture.
Cells communicate with one another by means of chemical signals. For the receiving cell, there are 3 stages in the signaling process: reception, transduction, and cell response. The cell targeted by a particular signal has a receptor molecule complementary to the signal molecule or ligand. The ligand fits like a key in a lock and triggers a change in the receptor molecule. Signal transduction converts the change in the receptor to a form that can bring about a cellular response. This might involve a series of steps, a signal transduction pathway that alters and amplifies the change. In the 3rd stage of cell signaling, the transduction process brings about a cellular response. This can be any of many different cellular activities, such as activation of a certain enzyme, rearrangement of the cytoskeleton, or activation of specific genes.
So the animation and talk just laid out this entire process for you. So figure 5.20, notice it says S-3 up here in the upper left hand corner. I removed the first 2, and I just went ahead and showed you what the whole thing looks like laid out. So you have the reception piece, you have a signaling molecule that binds to the receptor, which causes some like chain of events to happen. That's the transduction piece. And then finally you get the response that was desired in the end. So it's not such a direct thing like the local signaling. All right, so this is about the reception part of the long distance signaling. The first bullet gives you a new vocab word. So you might have heard him say ligand, which is not how I pronounce it. I say ligand, but just get used to it. It's the same word. So ligands or ligands, they're signaling molecules, and the receptor that they bind to are very specific. So in other words, they're not going to just bind with any molecule, they bond with some specificity. The ligand binds its specific channel protein, and then once that happens, the shape of the protein changes, and that is what activates this series of events. So that is the reception process. It's very simple. So there are two types of receptors we see over and over again, and they are G protein receptors and ligand or ligand-gated channels. So G protein receptors or G protein couple receptors, they're found in the cell membrane. They bind with substances outside of the cell, and then they transmit signals from whatever it is that they are binding with. So once the molecule, it could be a hormone or a neurotransmitter, once it stimulates the receptors, then that G protein is going to couple or bind with something called GTP to form an active complex. And an active complex just means that it's ready to go. An active complex that is actually going to help along other events that need to happen within the cell to get the desired response. So there are different types of G protein coupled receptors, and we see them in animals and in plant cells. There's some weird analogy out there, like a metaphor, that G proteins are kind of like a dancer, and it really just depends on their partner, the way in which they'll respond. So that's one way to think about them, because I did mention that, you know, it could be a neurotransmitter or hormone that binds with them, and depending on what it is, you may elicit a different type of response. So here in figure 5.21, I skipped the first one again and just went straight to this. So you have activated G protein coupled reactor. So that's what this, hold on one second, that's what this whole thing is here. Activated GPCR. And you notice that the signaling molecule has bound with it, which activates the G protein. And you see that to the right, there's an inactive enzyme. But once the signaling molecule actually leaves the activated GPCR, it allows GTP to go ahead and move, and it contacts the enzyme to the right that was once inactive. And once it makes contact, then that enzyme becomes active and you get a cellular response. So what's key here is that sometimes it's not the coming to and then binding of a receptor and a substrate or like a outside molecule. Sometimes what needs to happen is a molecule needs to remove itself in order for a reaction to go forward or for a response to happen. So this slide is about the ligand-gated ion channel proteins. They open and close just like a gate. So gated protein or gated receptor is appropriate. They open and close like a gate. They change shape with specific ions that bind with the receptors. What do you need to know? So sodium and calcium ions, they're transported into nerve cells via these channels. They're crucial to proper functioning of the nervous system. And the ions, they trigger those electric signals that we discussed when we were talking about how the neuron was working, moving that charge down the axon into that space that's in between the end of one cell and then the target cell, and then, you know, the signal continues to go forward. So here, figure 5.22. We're looking at an ion channel receptor from start to finish. So notice you have the ligand-gated ion channel receptor. That's a mouthful, right? There it is. Right now the gate is closed, but you have this ligand or the signaling molecule that's sitting right outside of it. As soon as it binds with the ligand-gated ion channel receptor, it opens up. And once it opens, just like the gate, then you can have those molecules flow in freely or out. It depends on whichever direction they need to be going. And then as soon as the signaling molecule removes itself, so when the ligand unbinds, then the gate closes right back up. And literally all it takes is for the signaling molecule to rebind, and then you have the same process that happens over and over again. All right, in addition to the receptors that are on or they're embedded in the cell membrane, we find that there's some receptors that are actually located in the cytosol, so they're inside of the cell. Those receptors, they're usually triggered by molecules that are hydrophobic. So because they're hydrophobic, they can easily slip through the phospholipid of the cell membrane. And there are examples given to you in the very last bullet point here. Note that steroids are listed, and what you should remember is that steroids belong to the class of macromolecules called lipids. Lipids, they don't mix with water. This is one way to remember that the molecules themselves are hydrophobic. Think about those long hydrocarbon chains that are associated with them. So there's your callback to chapter 2, I think. No, it was chapter 3. Talking about macromolecules again. All right, so this slide is going to walk you through the way something called aldosterone, which is a hormone, the way that it works. So what happens is the molecule comes from your adrenal gland and it flows through your blood, and then it goes really all over your body. But what's interesting is that the cells that it needs to target are actually in the kidney, and they're the only ones that have the receptors that are specific for binding to aldosterone. So once it lands in the right place, so once it hits the kidneys and reaches those cells, it will actually start a chain of events that ultimately will turn on a gene, or maybe even turn off a gene if there's a lack of. But that gene is responsible for the regulation of sodium and water flow into or out of a cell. So I'm simplifying here so that I'm not just talking about it. You can pause and read this in more detail if you like. All right, here is the image of what we just said. You see the hormone. It doesn't have any enzyme or any channel that it needs to slip through. It just comes right through the membrane. The receptor protein is inside in the cytosol or cytoplasm, the space in between the cell membrane and the nucleus. Once it binds, you get this complex that's formed, and what happens is it ends up being able to slip through a nuclear pore into the nucleus. It looks for a specific sequence within the DNA, and it basically transcribes it. It writes it into mRNA code. The mRNA will slip out of the nucleus, and then from there, the tRNA molecules, so that's what would be bringing the different amino acids to make this new protein here. They're going to go to work. You also notice that the ribosomes, there are 3 of them actually sliding along that mRNA strand to make the desired protein, which will ultimately do the ebb and flow of sodium and water into and out of the cell. So it sounds like something so simple, but it could get ultra complicated. Just keep in mind what the goal is. The goal is to create some type of cellular response. This just happens to be one example. All right, so what you may have began to notice is that the binding of a molecule to a receptor, it might start a signaling event, but the transduction that takes place, it can often involve a series of additional steps before we get to the end result, especially with what we just saw. So we call this a cascade. It's like a domino effect. One thing pushes the next thing, and so on and so forth until we get the desired end result. So transduction by cascade, it can be complex chemically. However, the pathways, they're not too hard to follow. The second bullet here is very important. It's saying that a few molecules can go a long way, and they can create a large response. And you want to keep that in mind. So just a few molecules can produce a large cellular response. And that's important. You want to be able to conserve our energy, and this domino effect transduction pathway can kind of help us with that. So this slide is just simplifying some of the things that I've already stated. It's restating them for you. I didn't feel like taking it out because I was like, oh, maybe they want to read it. So you can feel free to pause here to read further or just take notes for your own understanding. All right, so we're moving right along here. You may notice that there's some huge intimidating words at the very top of this slide, and they're laced throughout the slide, but don't be afraid. So their meanings are actually laced within the word roots, prefixes, and the suffixes, as always. You know how much I love a good word breakdown. So phosphorylation, what is it? It simply is adding a phosphate group to various proteins. So if we put D in front of it, so dephosphorylation is just the opposite. It's going to be the removal of a phosphate group. So protein kinases, they're enzymes. How do you know that they're enzymes, Ms. Johnson? Well, most enzymes, they end with this suffix ase, ace. So whenever you see a word that has an ase ending here, you're talking about an enzyme, which means that it's a protein, which means that that's the class of macromolecules it belongs to. So protein kinases, they're just enzymes. So they're going to speed up a chemical reaction, because that's what enzymes do. And the chemical reaction that they're doing is actually transferring phosphate groups from ATP. So what is ATP? Again, it's the energy currency of a cell. It's made in the mitochondria, so I can say that now with no hesitation, because you all know what the mitochondria is, because we've been talking about cells for forever at this point. But anyway, so protein kinases, they're going to transfer phosphates from ATP. They are doing the phosphorylation that we have talked about. When we encounter a pathway that involves the addition of a phosphate and then the cleaving off of a phosphate, they call this a phosphorylation cascade. In other words, the basic idea here is to remove a phosphate group and then maybe to add a phosphate group, or vice versa. That is a phosphorylation cascade. Bam. So the process there, it changes proteins from an active state to an inactive state, and it can do it over and over again as long as the proteins are not denatured. In other words, they're not unraveled. So here's a picture of a phosphorylation cascade, or rather a model. So you see at the cell membrane, so I'm looking right here, you've got your signaling molecule that is placed inside of that protein that's sticking out of the cell membrane. It's embedded within it, and once it's there, it activates another molecule. And then you have a series of events of inactive protein kinase turning into active protein kinase. And every time that happens, it is cleaving off one of the phosphate groups. So we go from having ATP to making ADP. And here's the phosphorylation part. We're moving that phosphate group from the first molecule of ATP to this second active protein kinase. At first it's inactive. As soon as it's phosphorylated, it becomes active, and the process just keeps happening. So we do it again, and then finally you reach the protein that you're trying to reach. It is phosphorylated, and then you get to your cellular response. Again, please do not be afraid of these words or how these diagrams look. Keep in mind what the goal is. It is just to signal a cell to get a desired result. This is just one of the pathways that you may encounter through this process. So now we're getting just like wordier and more word stupid, and I don't literally mean stupid for all of the cynical people who are out there listening, but the vocab here is just getting a little crazy. So protein, okay, this sounds ridiculous. Okay, protein phosphatases, it doesn't even sound real, but it's a real word. So protein phosphatases, they're enzymes, and they're enzymes that do dephosphorylation. Look at that. So in other words, they're proteins that remove phosphate groups from proteins. That's what they are. It ends in ACE, so I know it's an enzyme, and an enzyme is a protein. They turn off the signal transduction pathway. And that, by doing so, like, they help to preserve the protein kinases so that the same enzymes can be used over and over again to respond to signals like as needed. So we've established that the ligands, or the ligands, are the first things that we bind to the receptors, and then we call those the first messengers. This is the first time that they've used this for you all, but whatever, I'll teach you what it says. So first messengers, ligands, ligands, same thing. So from there, where do we go? So since we've talked about the transduction pathway involving a series of steps or a cascade, we should have no problem understanding that there may need to be a second messenger, and the second messengers are just as important as the first ones. So these messengers, they tend to be non-protein molecules, which is, I guess, important. They're usually water soluble. In other words, they break down in water. They could be ions, and they just kind of diffuse through the cell naturally. So we're not talking about any active transport or anything like that. They're just doing their natural thing. So two common secondary messengers that are worth knowing and that are listed here are in the last bullet point. So we have cyclic AMP, that's one. You'll see it written a different way on the next slide. Cyclic AMP, and then calcium ions. So those are some common second messengers just to keep in mind. All right, so you got the CAMP or the cyclic AMP already laid out. This next one is like just really ridiculous to try to pronounce, so please don't laugh at me, or just laugh at me, it doesn't matter. So it's adenyl cyclase. This YL here is like super weird to me, but anyway, so ASE, here's that ending again. It's an enzyme. It is in the plasma membrane, so we're inside of the cell at this point. What does it do? It converts ATP to CAMP. And why does it do it? Well, it gets a signal rather, and it's a response to this signal that happens in the extracellular space. And it's not just one. So according to this, there's a number of extracellular signals that would cause this conversion to happen. So it says that the immediate effect of the CAMP is usually to activate those protein kinases that we were talking about before. They list a specific one here, protein kinase A, so that's the first one. And then that will phosphorylate a bunch of other proteins. So we get that like a raining down of chemical activity that's going to happen in this transduction pathway via our second messenger. All right, so here we go again. Here's your G protein coupled receptor. Boom. You have the first messenger or your ligand/ligand. It's going to pull away. As soon as it does, you get the GTP moving. It hits this adenyl cyclase enzyme here, embedded within the protein. When that happens, it sparks off that ATP CAMP response, and it will basically cleave off a phosphate group. Protein kinase will continue to move that phosphate group until we get the desired cellular response. So we've covered reception and we've covered transduction. The only thing left is the response portion of this. So number one, what is the goal here? So the answer to that is in our first bullet. What we want to do is to basically carve out a pathway or spark a pathway that's going to lead to the regulation of one or more. So we're wanting to regulate one or more cellular activities. That is the goal. Here's the next question. Where is this happening? So our answer is in the second bullet. So the response, it might be happening in the cytoplasm, or it could be happening in the nucleus. Okay, so we've covered what's the goal? We've covered where it's happening. Now let's talk about the how of it all. How does this all work? So that's in bullet 3. So these pathways, they regulate the making of enzymes or other proteins. So that's what they're doing, synthesis of enzymes or other proteins. And how they do it is by turning genes on or off within the nucleus. So we kind of saw that when we were looking a couple of slides back. So this very last bullet point, the final activated molecule, so the last molecule that we turn on is the signaling path, in the signaling pathway. That is what is going to make that transcription factor. In other words, it's going to spark that protein to be made that will shut something on or off. All right, here we go. Let's look at the picture again. Keep looking at the picture. Study these models, guys. So you have the cell membrane right there. Boom. In it, you have the protein. You have, this time we're using growth factor. So they say growth factor is the molecule that's going to bind with the receptor. It's going to cause a phosphorylation cascade, and that happens with those whole protein kinases moving or phosphorylating and dephosphorylating things. So transduction happens there. Once that goes over, you enter into the nucleus. In this case, we have an inactive transcription factor. How do we turn it on? Well, it's going to be turned on by us adding that phosphate group from that phosphorylation cascade. That in turn will cause this particular molecule to read the DNA, and it will read for a specific sequence. It's looking for a specific gene to make a cell do something, to signal another cell to do something. In this case, it's found where it needs to read, which is the response itself, and you make this mRNA from the gene that's read, and that is going to make you a polypeptide, which will then be folded into a protein, and the protein will control something or make something happen. So other pathways, they do help in regulating the activity of various enzymes, and sometimes it's not always that you're needing to synthesize a protein. It could be some other things that you're trying to accomplish with the signal transduction pathway or just cell signaling in general. One of them that they're listing here is the opening of an ion channel, or to change the metabolism within a cell. In other words, to mess with the metabolic functioning or the chemical reactions that are occurring in the cytoplasm. So I'm done, and I'm done, and I'm done, and I will see you all tomorrow.